Now, in today's lecture, we're going to pick up where we left off with that discussion. Remember, our interest in the origin of life is that it's going to tell us something about how to define life. But where this material will take us today and in the next lecture is to begin to introduce the concept of information in living systems, framing the central theme for this first part of the course. The significance of Miller's experiment was simply to show that non-biological processes could result in the formation of organic molecules. As we'll see later in the course, actually starting in just a couple of lectures, polymers are essential because the most important biological molecules, especially proteins and the nucleic acids DNA and RNA, are polymers made up of different combinations of just a few monomer building blocks. Now, ordinarily, in living things today, there are a whole series of specialized proteins. We call these proteins enzymes, and we'll talk about that more later in the course. But these proteins acting as enzymes are what are responsible for building these polymers out of the monomeric building blocks. What happened on the early Earth in the absence of this specialized protein machinery that could possibly have led to polymerization? Could polymers also have arisen abiotically? Now, the first evidence that this was possible actually came fairly early on, again in the late 1950s, and it was work by Sidney Fox, who was at the University of Miami. Fox took Miller's experiment one step further. He was able to take, for example, amino acids that might have been created in an experiment like Miller's and get them to start joining together, but only under certain conditions. For a few kinds of amino acids, in just the right proportions, at just the right temperature, the right amount of time that you might heat them, Fox was able to do was to get fairly short polypeptides, showing that you can get spontaneous abiotic polymerization. The problem was that Fox could only do this under a very narrow range of conditions. In Miller's work, you could just throw a bunch of stuff into those flasks and you'd get some sort of organic molecules. But Fox's work required much more controlled conditions, conditions that are unlikely to have been found on the early Earth. But Fox and a number of other scientists speculated that maybe you could increase the hit rate. Maybe you could get more spontaneous formation of polymers if you had some sort of non-biological catalyst. What they suggested, thinking about the composition, the structure of the early Earth, was that perhaps there were certain kinds of clays that acted as inorganic, abiotic catalysts. Why clay? Well, it turns out that some kinds of clays, when they dry out, form very regular lattice-like structures, almost crystalline, not really crystals, but very regular structures. And furthermore, these clays will also have weak electrical charges on their surface. And these weak electrical charges can adhere organic molecules. Now, the idea here is that sometime in the early Earth, the shores of a primitive ocean had a bed of clay. And as the organic molecules that were being created in that primitive ocean got lapped up onto the shore, they adhered to that clay. And the clay, because of the regular order, the spacing, and holding those molecules in place, would increase the probability that you'd get some sort of spontaneous polymerization. Well, that's an interesting idea. Is there any evidence that this could work? I mean, we don't know what the primitive Earth was like on that scale, but it turns out that recent work by James Ferris, who's at the Rensselaer Polytechnic Institute, has shown exactly that this process does work under abiotic conditions in the laboratory. Ferris and his colleagues have been able to synthesize not only short polypeptides, but also short stretches of DNA from the component building blocks, amino acids on the one hand, nucleotides on the other, that were created from experiments like those done by Miller. We can postulate that biological polymers could arise spontaneously. Okay, so let's imagine. Let's imagine that we've got complex polymers. Let's imagine that we've got a primitive ocean brimming with a whole bunch of organic polymers, what's been called in the popular press the primordial soup. Where do we go from there? Let's imagine even that some of these polymers, by chance, have come together as strings that might even have some sort of useful biological function, like modern polymers. The experiments of Miller, Fox, Ferris, and others have shown that this is possible. But even with all of this, we still don't have anything approaching what we'd want to call life. Why not? Well, because we know that the organic molecules that make us up are not just a jumble of things floating around in a primordial soup. They're highly ordered. And specifically, they come in highly ordered packages. All living things are made of units called cells. We're going to talk more about cells in the next lecture. So minimally, for something to be living requires a barrier between the living part and the non-living part. And that barrier is what would define the outline, the barrier of what we might want to call the cell. Now, modern cells are very complex, and we're going to talk about those in the next lecture. But is it possible that some cell-like structure could have arisen, again, spontaneously on the early Earth? Here, too, laboratory experiments suggest that the answer is yes. I won't go into the details. There's been a lot of work on these. Are called early life studies. But a number of experiments have been done that demonstrate under conditions that are not too rigorous, the kinds of conditions that might have happened in the early Earth, you can get aggregations of abiotically produced complex organic molecules that will spontaneously form themselves into cell-like structures. So these kinds of spontaneously made cells in the laboratory are generically called protobionts. I'll use that term later. Protobiont literally just means, sort of first biological thing, protobiont. And you can make protobionts. It's actually not too difficult to do that in the lab. You can make them under a number of different kinds of conditions with a number of different kinds of organic molecules. For example, if you have the right kind of lipids, remember, those are the molecules that make up fats. If you have the right kind of lipids, you can almost literally just put them in water and they spontaneously form a package where there's a membrane of lipids that enclose some central space. Now, the most remarkable kind of protobiont, one that's been called a coacervate, is one that has been made to self-assemble out of a solution that includes polypeptides, short protein-like molecules, nucleic acids like DNA and RNA, polysaccharides, that's another term that we would use for polymerized sugars. Remember, the sugars are a group of organic molecules that we talked about previously. So if you get a bunch of polypeptides and nucleic acids and sugars together, and now you need to have the conditions a little bit more controlled here, you can nonetheless get these to self-assemble into a cell-like object. And what's really interesting about this work on coacervates is if you then throw into the mix some real biological molecules, for example, a protein enzyme that you've taken from a real living cell, a modern cell, and you extract that enzyme and you put it in with these coacervates. They're just in your laboratory dish. The coacervates can take up those enzymes. They'll bring them inside themselves. And in fact, those enzymes will start working inside the coacervates. So what enzymes do is they process one kind of biological molecule into another, and we would call what they're working on the substrate, and they produce some product. And so once these enzymes have been taken up by the coacervate, it will also start taking up the substrates for those enzymes, doing the reaction, and putting out the products. Now, we've given it the molecular tool, the enzyme, but nonetheless, we gave the coacervate the hammer, and it started hitting something. This is getting really remarkably close to something that we might want to call living.